Hi, my name is Steven Diaz. I am going to talk about cluster of Sphingomonas paucimobilis. Bacteremia lead to diversions of intravenous hydromorphs. Definition: Sphingomonas paucimobilis is a striline aerobic gram-negative bacterium that have a single polar filament with a slow motility. Sphingomonas species can cause hospital-associated death, infection from various mammimontal exposures, include reservoir with weakening the hospital plumbing.
Here you can catch the key idea of published research paper in less than 5 minutes. I hope it will help you to select the paper for your further reading, knowledge or research purposes. Viewers, today's paper is about mechanisms of CR4 resistance by endophytic Sphingomonas species LK11 and its CR4 phytotoxic mitigating effects in soybean. Saqib Bilal is the lead author. Viewers, chromium CR4 is highly toxic and leads to impaired phenotypic plasticity of economically important crops. The current study assessed an endophytic bacteria assisted metal bioremediation strategy to understand stress alleviating mechanisms in soybean plants inoculated with Sphingomonas species LK11 under severe CR4 toxicity. The screening analysis showed that high CR concentrations slightly suppressed LK11 growth and metal uptake by LK11 cells, while significantly enhancing indole 3 acetic acid IAA production. Endophytic LK11 significantly upregulated its antioxidant system compared to control by enhancing reduced glutathione GSH catalase CAT, peroxidase POD and superoxidase dismutase SOD activities to counteract CR-induced oxidative stress. CR toxicity induced cell morphological alteration as shown by SEM-EDX analysis and triggered significant lipid peroxidation. The interaction between LK11 and soybean in CR-contaminated soil significantly increased plant growth attributes and downregulated the synthesis of endogenous defense-related phytohormones, salicylic acid and abscisic acid by 20% and 37% respectively, and reduced CR translocation to the roots, shoots, and leaves. Additionally, CR-induced oxidative stress was significantly reduced in LK11 inoculated soybean, regulating metal responsive reduced GSH and enzymatic antioxidant CAT. The authors concluded that LK11 may be a suitable candidate for the bioremediation of CR contaminated soil and stimulation of host physiological homeostasis. Viewers, this article was published in December 2018 in the Journal of Ecotoxicology and Environmental Safety. Viewers, I hope you found this video useful. I would really appreciate your feedback. Please don't forget to subscribe and follow the channel. Thank you.
Application in medicines. Sphingomonas paucimobilis, a non-fermenting gram-negative bacillus, is regarded a minor clinical significance. Infections include Bacteremia, Septicemia, caused by contaminated solutions like distilled water, hemodialysis fluid, and sterile drawn solutions.
Today's video, I just want to very briefly talk about the main differences between gram-positive and gram-negative bacteria. So these two diagrams here show you a very simplified version of gram-positive and gram-negative bacteria. If we look at the internal structures, they're very similar. We have this DNA strand here in both of them. We have ribosomes dotted throughout the cytoplasm. And we have this circular DNA, which is also known as a plasmid. I want you to look at the outer surfaces, and this is where we have the key differences between both types of bacteria. The gram-positive bacteria just have a cell membrane and a cell wall which is made from peptidoglycan, and then they have this periplasmic space which just separates the two. Peptidoglycan is what the cell wall is made from, and it's just a combination of sugar and amino acids, and it forms a meshwork like this. In gram-positive bacteria, this layer is very, very thick. In gram-negative, we have this cell membrane, we have the cell wall made from peptidoglycan, but it's much thinner in comparison. We have two periplasmic spaces, and then we have an outer membrane which is made from liposaccharide. So gram-positive doesn't have this additional layer, the outer layer is just the cell wall. However, gram-negative has an outer membrane made from liposaccharide. The reason why this is important is because when you have a sample of bacteria and you want to know which antibiotic to use, the antibiotics all have a different mechanism of action. So one antibiotic which works on gram-positive bacteria might not work on the gram-negative bacteria because of this difference in their structures. So this is why it's very important to know the type of bacteria that you are treating and whether it's gram-positive or gram-negative. So an example of gram-positive is Clostridium, an example of gram-negative is Helicobacter. I'll talk about this staining technique as well, which I don't want to go into too much detail about, but I'm going to use this video sample here. I'm going to explain what's going on. First, what he's going to do is take his bacterial sample and spread it across the microscopic slide and then heat the bacteria in place just by dragging the slide over the flame very lightly, and the bacteria gets fixed in position. And then he's gonna place this liquid over the slide which is called crystal violet, and it's got this purple color. Then he's gonna wash off this crystal violet after leaving it on for a minute. Then he's going to place this grams iodine on it. So it's basically like an iodine solution which is going to be placed on the sample and left for a minute, and then it's washed off. Now the gram stain or the purple stain color is going to be in the cell membrane and peptidoglycan layer in both gram-positive and gram-negative bacteria. But now we're going to wash away the gram stain in gram-negative bacteria by using ethanol. So what ethanol does is it dissolves the outer membrane, and since the peptidoglycan cell wall is so thin, this is going to get dissolved away. So essentially the purple stain gets removed as well. However, in gram-positive bacteria, since the cell wall is so thick, the stain remains in gram-positive. That's why the gram-positive stain purple. Finally, we can stain gram-negative bacteria using safranin, which stains the gram-negative bacteria in a reddish pinkish color. So this safranin does not stain gram-positive bacteria, it only stains gram-negative bacteria. So the overall effect of the gram staining technique on both gram-positive and gram-negative bacteria, it has this final appearance in the microscope image. So lastly, just to end the video, I want to make a few final points on the differences between gram-positive and gram-negative bacteria. I've already mentioned the first 3 points in terms of the gram staining and the differences in the membrane structure. The fourth point is that gram-positive bacteria have teichoic acid and gram-negative bacteria do not. Teichoic acid is a glycopolymer and is responsible for bacterial cell division, and it also helps to maintain the shape. And the final difference is that there is very little liposaccharide content in gram-positive bacteria, and there is a much higher content of liposaccharide in gram-negative bacteria.
Pros and contra. Pros. Sphingomonas species rarely cause bloodstream infection even among immunocompromised patients. Is regarded as a minor clinical significance. Cons: created its significant problem in clinical settings, being the most widespread cause of monosocomial infection, central catheter-related bacterial, peritoneal catheter-associated peritonitis, meningitis, and other infections. Include plant in Ecuador. The organization is bile distribution in soil and water, including water sources in the hospital environment. They are opportunities pathogens that take advantage of underlying conditions and diseases.
Welcome back. In this video I'll be teaching you everything about Pseudomonas aeruginosa that you'll need to know. So to memorize all the features of Pseudomonas aeruginosa, I use the mnemonic Pseudomonas. It's the same mnemonic that a lot of people use, but I just modified it a little bit so it contains more information and it's easier to remember. So P stands for pneumonia, especially in cystic fibrosis patients. And S stands for sepsis. E stands for ecthema gangrenosum, which is a rapid skin necrosis in immunodeficient patients. And U stands for UTIs. D stands for drug abusers and diabetic patients, which are common targets of Pseudomonas aeruginosa. O stands for osteomyelitis. M stands for mucoid capsule and melted skin infection, which is infection of melted skin in burn patients and burn victims. And the second O stands for otitis externa, which causes swimmer's ear syndrome. N stands for nosocomial infections, especially those patients with long-dwelling catheters. A stands for all toxins of the bacteria, and they are exotoxin A, which will inhibit protein synthesis of the host, endotoxin, which will cause shock, phospholipase, which will cause cell lysis, and pyocyanin, which is a toxin produced by Pseudomonas to kill competing bacteria. And finally, S stands for skin folliculitis, which is also known as hot tub folliculitis. Other features of Pseudomonas is that it causes blue-green pigmentations if it's going to cause any pigmentations, and also causes keratitis in patients wearing contact lenses. And to treat Pseudomonas, we use anti-pseudomonal beta-lactams like penicillins and cephalosporins, and we also use aminoglycosides and carbapenems. Alright guys, so that's everything I've got. Hopefully I made this easier for you, and see you guys later.
Conclusion: Sphingomonas paucimobilis is a non-fermenting, aerobic gram-negative bacillus that may cause both monosocomial and community-acquired infection, albeit rarely. It has been reported to proliferation in distinct water, hemodialysis, fluid, and sterile drug solution. Thank you.